Hey everyone, welcome to week 2 of our online lectures. I ended our last video, last section, talking about the evolution of vascular tissue and some of the life cycle evolution in the vascular plants. And today, so that just as a reminder, that vascular plants included our lycophytes, or club mosses, and the ferns, the Manilophyta. And I talked about different lycophyte groups and different fern groups, and we went through the evolution of some of those characteristics, including the evolution of vascular tissue and of leaves, both microphylls that we see in lycophytes and megaphylls that we see in ferns. Today what I want to do is finish out the vascular plants and actually finish up the plant diversity and evolution section through this last video and move into our last group. And so to do that, I want to move into our last group of plants, the spermatophyta, or the seed plants. So sperm means seed, just like all of these phyta as plants. So these are our seed plant lineages. And I want to use this slide to point out, while we've been talking about vascular plants contain the majority of our diversity, but that diversity is very unevenly split. And so in our vascular plants, about 1,000 species are lycophytes, around 12,000 species are ferns, and then over 250,000 species are seed plants. And so the majority of our plant diversity is actually in this seed plant group, which I want to move into now. And so looking at our vascular plant phylogeny here, that we went through most of these characteristics: sporophyte dominance, having vascular tissue with both xylem and phloem, xylem moving the water, phloem moving photosynthate. We have the evolution of megaphylls. Go ahead and change that here. Megaphylous leaves, and we talked about microphylls in the lycopods, whereas megaphylls are in both our fern groups, including the whisk fern, Psilotum, and the horsetails, Equisetum, in our ferns, and then our seed plants. And so all of those have megaphylls. What I want to focus on today is the evolution of two other major morphological features that we see in vascular plants, and specifically in seed plants, and that's the evolution of both wood and seeds. And so wood we'll have a very specific definition for, and I'm going to move into that one first. As we look at our vascular plant lineages, just like we've seen before, we see the group of lineages leading up to our seed plants, or the seedless vascular plants, are paraphyletic. So these are not a monophyletic group. Seedless vascular plants themselves are paraphyletic. We already knew that with our lycopods and our fern groups, the ferns and horsetails being those first two lineages. But what I've included in here again is a couple of fossils, the Anangiosperm and Archaeopteryx. These are two fossil groups that we call, not extant today, but we call both of these progymnosperms. And what I want to point out here is that both of these fossils are woody fossils. They contain wood. And so wood has, like I said, a very specific definition, and that is being secondary xylem. And that secondary xylem is produced from a cambium. And I will explain both of these characteristics. I'll come back to heterospore and seeds. So I first want to start out with wood and the evolution of wood and how we see that evolve in these progymnosperm groups that are related to and share a common ancestor with our extant seed plants. Okay, so let's take a look at some of these fossils. This is Archaeopteryx. I mentioned this is a progymnosperm. And so it has a lot of the features that gymnosperms have, but one of the things it does not have is seeds. So these are actually reproducing more similar to what our seed-free or our seedless vascular plants, like our ferns, our extant ferns and lycopods, reproducing by spores, not reproducing by seeds. And so these are, like I said, woody plants, but they do not have a seed. So in those progymnosperms, we actually can look in the fossil record and find fossils of these that includes large trunks, like this fossil trunk here. And if we look at this under the microscope and look at a section through the vascular tissue, we actually see wood here. And so these fossils contain wood. And what I mean by wood is specifically it contains secondary xylem. And I'm going to go through what primary xylem is and what secondary xylem is and how we actually differentiate these. But because they contain wood or secondary xylem, these actually are large trees. And so this is an important step because this now having vascular tissue and having secondary vascular tissue, secondary xylem, allows these to get to be much larger plants. So prior to this, our plant groups are relatively small. They don't contain this large structural woody tissue that allows them to get very, very large. So how do plants grow? To talk about what secondary xylem is, we need to talk a little bit about how plants grow. In the next chapter, we're actually going to be delving deeper into this, and so this will kind of be an introduction to some of these terms. But in order to understand what secondary xylem is, we first need to understand the primary tissues themselves. And so plants grow from meristems. Meristems are undifferentiated, perpetually young plant tissue from which new cells arise. So all new cells in a plant come from a meristem. And we have a couple of different types of meristems, both primary meristems and secondary meristems in a plant. What I'm showing you here in this image is actually the image of a primary meristem, and that is the apical meristem. So the apical meristem is where primary growth occurs in plants. And so what this is growth in height or length in the plant. And so what we're looking at here is actually a cross section through the very tip of a plant stem. And if we look at that cross section of the very tip of the plant cell, we have this of a plant stem, we have this group of cells right out there at the tip that we call the apical meristem. So these are perpetually young, undifferentiated cells, and it's the point of where all cell division is occurring in the plant, where cell division occurs. So in a plant, all cell division occurs in a meristem, and that's the point where cells are actually dividing. And so as these cells are dividing, the plant is getting longer or growing in height. And so primary growth in plants is growth in height or in length if you're talking about a lateral branch, height if you're talking about that main stem branch. So all of that is happening in that apical meristem. And so if we look at what's going on with the rest of this image here, we've got a few different things. And so these are leaf primordia. So as those cells get older after cell division, they sort of migrate down the apical meristem and they can start to differentiate into different types of cells. Some of those cells are going to be morphological features like the leaves of a plant. And so we'll end up differentiating into the leaves. And so those surrounding the apical meristem there, these are our leaf primordia that are formed. So these are young leaves that will develop into those mature leaves that we see on a plant. We also, as those cells differentiate, they'll differentiate into vascular tissue. So we see actually some vascular tissue running into these leaf primordia and vascular tissue running down the stem of the plant. So this is all vascular tissue, xylem and phloem, produced by the apical meristem. So because it's produced by the apical meristem, we call that primary vascular tissue. And we also see a couple different things here on both either side here in the axil or adjacent to the stem in the axil of the leaf, we see axillary meristems. And so these axillary meristems will grow into a branch. And so at the end of those branches, we have an apical meristem, just like we see on this main axis of the plant. And so the tips of all of these grow is where the growth of a plant happens. So plants all grow from the tips, and that's that primary growth coming from those apical or axillary meristems. So cells dividing and then differentiating into the different parts of the plant. Well, also in a plant stem or in some plant stems, we have other types of meristems. And the one that I want to mention here with respect to wood is the vascular cambium. The vascular cambium is a secondary meristem. And so this is where secondary growth occurs here. And when I talk about secondary growth, what I'm talking about is growth in girth in this case. And so secondary growth, primary growth in a plant is growth in length. Secondary growth is actually growth in girth of that plant. And that's the result of division, cell division happening at that secondary meristem. And so what's the vascular cambium is a type of secondary meristem. I'll talk about why we call it bifacial in just a second. But the vascular cambium itself is a group of undifferentiated cells. So it's a meristem, undifferentiated cells that are actively dividing. And so in this drawing, in this image that I have here, where I'm putting the blue line, this is the vascular cambium itself. So this is that meristem, that secondary meristem. And so what, just to define the vascular cambium, this is a cylinder of cells that divides to produce secondary xylem, so to produce secondary vascular tissue. To produce secondary vascular tissue. Remember, vascular tissue in the plant is made up of both xylem and phloem. And so the reason this is called a bifacial secondary vascular cambium or bifacial secondary meristem is that this produces secondary xylem to the outside, to the inside of the stem. And it produces secondary phloem to the outside. And so as a result, the cambium is dividing and it's putting new cells to the inside of the stem, and it's dividing and putting new cells to the outside of the stem. And so basically you should picture a bunch of dividing cells, some that are dividing to the inside and some that are dividing to the outside of the stem. And that's why we see these new cells or this secondary xylem being formed in these nice, neat rows headed toward the inside. and same with the secondary phloem to the outside. It's because these cambium cells are actually dividing and putting a new cell to the inside, dividing and putting a new cell to the outside. And so these are bifacial. They divide on each of those faces, one to the outside and one to the inside of the stem, to create both secondary xylem to the inside and secondary phloem to the outside. So when we look at this, all of this tissue to the inside of the vascular cambium is our secondary xylem. All of this tissue to the outside is our secondary phloem. And it's secondary xylem that we call wood. So wood is actually made up of secondary xylem in the plant. That is what wood is. And so to be able to be a woody plant, you have to have a vascular cambium that produces secondary xylem toward the inside. And so here we see this in a young stem that has just started, where the vascular cambium has just started dividing. So this is the vascular cambium, and each of these is a vascular bundle. And this vascular bundle is made up of both primary xylem and primary phloem. So those are the xylem and phloem that were produced by the apical meristem, or the primary meristem. The vascular cambium, which is that secondary meristem, is dividing to produce secondary xylem to the inside and secondary phloem to the outside. And what we can see in this diagram is just the beginnings of that secondary xylem and phloem starting to be produced and separating those primary xylem and primary phloem from each other. The primary phloem is getting pushed to the outside, the primary xylem is getting pushed to the inside. And so I mentioned this here just to talk about wood. So wood is the secondary xylem produced by that vascular cambium. We will look back at primary growth and secondary growth in the next section of the course, in the next chapter, and we'll look at this again. And so this is kind of a primer for you, that we have woody plants that have a vascular cambium. That vascular cambium produces the secondary xylem that we call wood. Okay, so that gets us through our wood. That gets us through the synapomorphy of wood for our seed plants. And now I want to talk about the synapomorphy of actually having a seed. And so seeds in plants, to do that, I first want to remind you that seed plants are heterosporous. And so I have that on the slide here. We have these heterosporous seed plants, including Archaeopteryx. Archaeopteryx is that extinct plant that I showed you before. Archaeopteryx was heterosporous, but it did not actually produce seeds. So it was a heterosporous plant that reproduced by spores. In our seed plants, we also have the evolution of seeds. And so all of the seed plants are heterosporous. And what that means is that they produce two types of spores after meiosis. And so meiosis results in both the microspore, production of a microspore, the male spore, and a megaspore, the female spore. And just to remind you that in these two types of spores, so heterosporous is two types of spores, and the gametophytes themselves are endosporic. And so just a reminder of what that means that these microgametophytes, the male gametophytes, and the megagametophytes, the female gametophytes, are actually developing inside of the spore wall. And so in seed plants, both the megaspores and microspores being endosporic, this is actually sort of the ultimate reduction gametophyte part of the life cycle of the gametophyte generation. So ultimate reduction of the gametophyte phase. And in some cases, as we'll see, this actually gets reduced down to just a few cells, but it's still a multicellular gametophyte. So we still have that alternation of generations between the haploid gametophyte generation and the diploid sporophyte generation. But because these are heterosporous and have endosporic gametophytes, these are very, very much reduced. I will, the last section of the course, we're actually going to go through angiosperm reproduction in a bit more detail. And so here I'm just doing this in sort of an overview to talk about the evolution of the seed. So just like with vascular tissue or with a lot of these characters, we'll go into a little bit more detail on them in later sections. But I just want to look at sort of a evolutionary progression here of sporangia, and what this looks like in seed plants. And so this is what we would see in a homosporous plant. So that just has one type of spore. In homosporous plants, we just have that one spore type. This would be a heterosporous plant where we produce both megaspores and microspores. So now I've got separate male and female gametophytes that are developing in this heterosporous plant. So this is something that we might see in like that Archaeopteryx, that's pre-seed plant. We don't have seeds yet, but we are heterosporous and produce both megaspores and microspores that develop into the megagametophytes and microgametophytes. Later on, through this progression, we actually see sort of the evolution of the seed specifically, in that seed become in seed plants, we end up reducing down to just one megaspore. And so our other three megaspores have degenerated here, and we have one megaspore that's sort of taking over. And in seed plants, the seed, when it's immature, we call it an ovule. This is an immature seed, so before fertilization occurs, we just call it an ovule. And all that is an integumented megasporangium. And so the megasporangium, that's the casing that includes the megaspores. So here's a megasporangium. And if we have that megasporangium that's reduced down in seed plants to just that one single megaspore that produces, in this case, an archegonium, it has the seed itself just has some protective layers around it. And so we end up with sporophyte tissue that's actually surrounding the megasporangium, the gametophyte tissue, where those spores are produced, and we're surrounding it with some protective layers. And we've seen this sort of change through the fossil record. And so all of these are interpretations from the fossil record of various levels of integuments, or these protective layers surrounding a megasporangium. So inside of each of these is a megasporangium with those megaspores inside. And the megaspore, remember, that female gametophyte will develop inside of the megaspore. And so this, on the inside here with the archegonium, is actually the female gametophyte of a seed plant. It's endosporic, meaning the gametophyte has developed inside of the spore wall, inside of the megaspore wall, and it's surrounded by those protective layers, which we call the integuments. And so those protective layers are the integument. And those are sporophyte tissue that's surrounding that, that mature female gametophyte. So seeds are just these integumented megasporangia. And so in angiosperms, this actually ends up being, I'm not going to go through too much detail here. As I mentioned before, we'll talk more about angiosperms. But in angiosperms, you end up with the ultimate reduction, or the ultimate reduction. And that is that we've lost archegonia. So angiosperms no longer have archegonia. They form their egg cells outside of archegonia. And so this is really the ultimate reduction of that gametophyte stage. And like I said, we'll talk a bit about that soon when we talk about angiosperm reproduction in general. In gymnosperms, we still have an archegonium or archegonia, multiple. And so here we have the archegonium. They're more simple than that sort of long, the urn and long neck shape that we see in other seed plants, but they're still an archegonium. So they have the sterile jacket layer surrounding the chamber where the egg is produced. Once that and we still have sperm nuclei that have to make it to the egg nucleus to be able to fertilize and then switch to the sporophyte generation. And so in gymnosperms, we still see archegonia, but in angiosperms, we've lost those archegonia altogether. Okay, so those are our two synapomorphies for seed plants. Wood, formed from, that is secondary xylem, formed from the vascular cambium, or that secondary meristem. And then seeds, which are actual integumented megasporangium, where once fertilization occurs, we can develop this embryo, which, remember, the embryo is sporophyte. So this is now sporophyte generation that is sort of in that seed. So the seed, it has the embryo all developed and ready to go for that next generation, but it's a sort of a pausing point in our sporophyte generation. A lot of things can stay dormant as a seed for a long time, but inside there is a little embryo, which is sporophyte, ready to grow out of that seed. So seeds are that second synapomorphy for seed plants. Okay, so let's take a minute and take a look at what a typical seed plant life cycle looks like in a little bit more detail. But you can guess where I'm going to start with this life cycle. I'm going to start with meiosis and that transition from the sporophyte generation to the gametophyte generation. And so when we start with meiosis, we've got, remember, these are heterosporous, so we've got actually 2 points in the meiotic. We have meiosis in both the female and meiosis in both the male side of this. And so we have in the male side of this, we produce our microspores. And the microspore develops into the mature male gametophyte, which is a pollen grain. So the mature microgametophyte is a pollen grain. So the outside of that pollen grain was actually the spore wall, that microspore wall. And then we've had a couple of cell divisions to create the male gametophyte, that mature microgametophyte, which is inside of that spore wall. And what we call that is a pollen grain. So pollen is the microgametophyte in gymnosperms and in angiosperms. Pollen is how the male gametes are being delivered. And so that's through pollen. In the female side, we actually have the production of our 4 megaspores. One of those is maintained. The other 3 products of that meiosis die. And so from that meiotic event, instead of having 4 megaspores now, we just have 1 megaspore that continues on. That megaspore divides to form the mature female gametophyte. And so the mature female gametophyte still has, so that's here. Here's our mature female gametophyte that has archegonia, and that archegonia has the egg cell inside of it, and that egg cell needs to be fertilized by a sperm cell. And so here's where in seed plants, the microgametophyte and megagametophyte need to come together, and that happens through pollination. And so in conifers, wind will move the pollen near the site of the female gametophyte, and so that pollen grain will land near that ovule, near that megagametophyte, and the sperm nucleus will be delivered to the egg nucleus. This happens in a couple of different ways. I'll show you in a couple of gymnosperm groups, they still actually have motile sperm that swim. Most of these don't. Most of our seed plant groups don't have motile sperm, but a couple do, and that's just amazing. But once that sperm and egg fuse, then we have transitioned we've transitioned from the gametophyte to the sporophyte generation. And so you'll notice that the seed itself, a mature seed that contains an embryo, is part of the sporophyte generation. So that seed, after fertilization occurs, the zygote is there. The zygote can start dividing and form a mature embryo. And then that mature embryo stops, and it stops growing, and it's contained within the seed wall, and that's a little dormant phase of the sporophyte generation. Once that germinates, it can develop into a seedling and developing sporophyte, and then finally into a mature sporophyte. It's in that mature sporophyte where our gametophyte generation starts to develop. And so the reproductive organs in a conifer, in this case, are both male cones, so pollen cones, the male side of things, and female cones, or ovulate cones, the female side of things. And if we open those up and dissect them, we see that there's actually little sporangia in the male cone, little sporangia, little balls associated with the little yellow cone scales, and that's where the microspore is going to develop inside those little microsporangia. On the female side of things, if you look at a cone scale itself, on the surface of the cone scale, we have megasporangia that are starting to develop. And that's where meiosis will occur inside the megasporangium to then create our megaspores and end up going back into the gametophyte part of this generation. And so the main thing that I want to emphasize with this is that the sporophyte is dominant. The gametophyte is much reduced. So a highly reduced gametophyte, and it's dependent on the sporophyte for nutrition. So we've flipped all the way full circle from our bryophyte groups that have gametophyte dominant and sporophyte dependent to now having sporophyte being dominant and gametophyte being dependent. Okay, what I want to do for the end of this lecture is just so you have a chance to see them. When we look at our seed plants, we see, or our green plant phylogeny, we see that our seed plants originated here. And so this is where we see both the evolution of seeds and the evolution of wood in both of these. And they consist of seed plants, extant seed plants consist of 2 groups. Gymnosperms, which are about 1,500 species, and angiosperms, which are probably greater than 300,000 species. So just like we saw with non-vascular plants and vascular plants, we see sort of unequal levels of diversity throughout. Within gymnosperm, but within seed plants, we have these 5 main lineages of seed plants. 4 of those are part of the gymnosperms, and the other one is the angiosperms, where we see the majority of the diversity. What I'd like to do at this point is to just walk through what these different lineages look like, and some of the main characters, just so you've at least seen some pictures of some of these. In lab, we would have had set up separate stations for gymnosperms that showed a lot of this diversity, and angiosperms, and seedless vascular plants, and non-vascular plants, showing what a lot of the major morphological characters look like. I want to take a few minutes now and just walk through some pictures of these major seed plant groups that we see, since they are quite diverse in their morphologies and what they look. And you've seen most of these before, and you may not have recognized exactly what they were. Okay, so the first of these gymnosperm groups is a cycad, and so I'll just note, I'm not going to take a lot of notes on these because I have most stuff written out, but this is a gymnosperm group. Gymnosperm, just so that we're all on the same page here, means naked seed. And what that means is that the seed is actually not contained within a fruit. The seeds are out there. Typically, these are in cones, but the seeds themselves will be produced on the surface of the cone scale and released into the environment, where angiosperm means clothed seed. And that's because the seeds are inside of fruits. So think about your apple, think about your cherry, think about any of the fruits that we eat, think about your tomatoes. Those all have seeds on the inside. Those are angiosperms, seeds on the inside of a fruit. So those are clothed seeds. So all our gymnosperm groups have naked seeds. The first of these are the cycads. Cycads are very palm-like or fern-like plants. Oftentimes they are mistaken for palms or ferns when you see these growing in the wild. There's about 130 species of them, and the seeds are very brightly colored and sometimes even have a fleshy outer layer to them. But these are common throughout the tropics and subtropics. One species is native to North America, and it's found in Florida, but it's planted all over the place. And that's this thing here, actually. This is Cycas revoluta. This is a common ornamental plant where it doesn't freeze. These things are oftentimes planted. And I just want to show you a little bit of the diversity. So these, like other gymnosperms, produce cones. And remember, these are heterosporous, so they produce female cones, or megasporangia. Megasporangia, and then also male cones. And so this is the male cone of a cycas, where this is the female cone. Just to give you an idea of the size of these things, some of these male cones can get to be a foot or more long, and these female cones are these big balls. And if we were to these are cones, they just don't have like our woody cone scales like we think of in a conifer. Here you actually have one of those cone scales, or sporophylls. And on there, these are the ovules that will end up being fertilized and producing the seeds at the base of those. Okay, so around 130 species or so of cycads. They're one of the most diverse groups outside of conifers of our gymnosperm lineages. Ginkgo is the second of our gymnosperm lineage that I want to talk about. Ginkgo is an interesting one in that there's only one extant species of ginkgo. Ginkgo biloba, which most of you have heard of, is the one that's left. There are fossils that go back nearly 300 million years of ginkgos, and there's ginkgo diversity that's beyond just this one species. So there used to be more species of ginkgos than we have today. All morphologically pretty similar. Ginkgos have separate male and female trees, and if you ever find a female tree, there's one on the admin lawn and there's a couple in the arboretum. They produce these you know, ping pong ball size or so seeds that have a very fleshy outer seed coat on them, and they stink like vomit. They smell like kind of a mixture between rotting cheese and vomit. They're really interesting. And inside of it is one big, inside of this seed with the fleshy seed coat is the, there's a hard sort of inner seed coat on there that gets dispersed. So yeah, they're cool. But they don't smell good at all. One of the things that I want to mention with this, both cycads and ginkgo, these are our two groups of seed plants that still have motile sperm. So here we see a sperm cell that has its cilia or its series of flagella on it, and these actually are motile and they swim to the egg. So these have pollen grains. Pollination occurs in the same way, where pollen grain will blow toward the ovule. Instead of the pollen growing a pollen tube and delivering the sperm nucleus, that pollen grain actually breaks open and the sperm swim out of it and then swim down to the archegonia. Okay, our next group of conifers, our next group of gymnosperms is the largest of the gymnosperm groups, and this is the conifers. The majority of the diversity in gymnosperms is actually found in conifers, so around 1,000 or more species of conifers. And these are the ones that we're most familiar with, ranging from our giant sequoias to our pine trees and junipers and yews and all of the different conifer groups that we're more familiar with. So 500 to 1,000 species of conifers. These are big woody plants, often needle-like leaves, and then the reproductive structures are woody cones that we're more familiar with. Okay, there are three different groups of there are three genera in a group that we call the Cnidophytes, and so this is the fifth of these lineages. These are weird plants that were previously very hard to classify. They share some unusual features with angiosperms in terms of their pollen morphology, some specialized vascular tissue, some details of fertilization are very angiosperm-like. However, these are more closely related to other gymnosperm groups than they are to angiosperms. There are three distinct genera in this, and I'll talk about each of them: Welwitschia, Needham, and Ephedra. Welwitschia is also commonly called the plant from Mars. And this Welwitschia, it grows in the deserts of Namibia, and that's the only place it is. There's just this one species. And it has these two leaves that grow throughout its lifetime. And so here's those two leaves starting out young. And then an older plant shown here where those leaves are all shredded out, but they just have these two leaves that grow throughout their lives. Needham is a group of mostly tropical trees, about 35 species. Here is a female cone of Needham. We see that it's not very woody cone-like, but we have our little ovules produced out here, and you can see a shining, glistening little pollination droplet there, and that's actually to catch the pollen from the male cone, which is shown here. That pollen will be produced and then caught in those little pollination droplets so that it effectively delivers the sperm to the egg cells. And then finally, our last one is Ephedra. There's about 35 species of Ephedra, and these grow, these are actually pretty common in our desert southwest. This is where the alkaloid ephedrine, which is a vessel constrictant, actually comes from. This is also the main ingredient of Mormon tea. And so growing in the desert southwest, these alkaloids that are being sort of utilized in this plant. So this is like low-level speed, basically. Ephedrine comes from this plant, Ephedra. And these are the female cones here and the male cones here. And then finally, our last group of seed plants are the angiosperms. Angiosperms are the most diverse of these, as I mentioned before, over 300,000 species and a huge amount of morphological and ecological diversity. I'll say morphologically and ecologically diverse.